Welcome students. Today we are going to have a very important class for the 5th semester student, a pharmacy student, and it will be interesting for other students also, pharmacy students. And this one we are going to have a class apart from the 5th chapter. Regarding about the extraction, isolation and characterization, in which I have taken to discuss about chromatography, and then there is various methods of chromatography, various principle behind we have to discuss. Today actually we are going to discuss about the definition for chromatography and the basic principle, and this first only one technique like column chromatography. We have very important thin layer chromatography, paper chromatography, HPLC, GC, all these things we have to come across in this chapter. Today we are going to discuss about chromatography, and in which we are going to discuss only one type, that is column chromatography. We will continue in the coming classes. Actually, in this chapter we have to discuss about extraction, isolation and characterization technique. These three words, even in the classroom I have told you. Extracting the active principle, extracting the medicinal active compound from the plants, and then isolating them by means of various techniques. By chromatographic technique is a very interesting technique, and afterwards isolating characterization technique by means of various instrumental analysis, including IR spectroscopy, NMR spectroscopy, mass spectroscopy, emission spectroscopy and UV. These all instruments we will have to know about the application in characterization of the structures. Today, in this isolation and characterization technique, what all the techniques we are going to see in this chapter is chromatography, in which column chromatography, paper chromatography, thin layer chromatography, high pressure liquid chromatography, HPLC, in some books high performance liquid chromatography, gas liquid chromatography, gas solid chromatography, GC is also available. Gas liquid chromatography usually we are using along with mass spectroscopy, GCMS, for plant constituents identification, phytochemical investigation, characterization can be done. High performance thin layer chromatography, HPTLC, is an important technique, very interesting technique, more useful for the research in herbal and natural sources. Then for characterization we have to study about the applications of spectroscopic studies in this chapter, like UV spectroscopy, IR, that means infrared spectroscopy, mass spectroscopy, nuclear magnetic resonance spectroscopy, NMR, and then other spectroscopies are also there, emission spectroscopy and various other detectors also there, flame photometer, turbidometry. These all you should know how they are utilized for the identification, characterization of the chemical structure of the compound which we have isolated from the plant source or the natural source. Then electrophoresis. You might have heard about fingerprint, for DNA fingerprinting, all these things from by gel electrophoresis, that is paper electrophoresis, gel electrophoresis, capillary electrophoresis. All these things we should know about the application towards the isolation and characterization of phytoconstituents from herbs. And here today we are going to see what is a chromatography. In very simple, the student should know that instead of mug-upping, we should know that chromatography is a technique which is used to separate mixture of compound. Any student can write. Chromatography is a technique which is used to separate mixture of compounds. So here chromatography is widely used for the separation, identification of compounds of a mixture. Chromatography is also very suitable for the extraction of plant extracts or isolation of plant extract, especially when our small amount of material is available and when the components are very close, resemble each other, slightly different in the structure or slightly different in the physical character, that can be easily separated, that can be very minute separation can be done by chromatography. Example, alkaloids, anthraquinones, quinones, alkaloids. These all are having a structures and the characters nearby can be separated even by chromatographic techniques. In the earliest form, that means the previous form, initial identification in 1960s, when they identified the chromatographic technique, was applied only on a colored compound. When they get separated, orange, red color, brown color, green color, like that colored component they got separated in a column or in a medium which they have kept. And the result of separation could be followed by visual observation. Because the color which we can identify, by that we can say this is this compound, that compound. That is the initial stage they made. As shown in the picture, you can see even in the paper chromatography, when the movement of the mobile solvent in the paper in an ascending manner against the gravitational force. In column chromatography, you can see the second picture, column chromatography, a column like a burette, a packing of material where the sample is kept up and getting separated. Color difference, you can see that orange, yellow, red, white, and a pure yellow color like that, you can see that. So colored substance they have identified previously initial stage. Just an example. So in chromatography, some classification, there are many types of classification. This is a simple by means of basis of their interaction to the solute to the stationary phase. That means the sample reacting with the stationary phase in what principle? By that you can have a classification. On the basis of chromatographic bed shape, that means what is the shape? Whether it is X and Y only or if it is three-dimensional like column chromatography, X, Y and Z. So it is not by means of the movement of the sample. There is two-dimensional, three-dimensionals work is in paper chromatographic and thin layer chromatography then can be done. That is different performance chromatography. But here on the basis of the bed shape, thin layer, paper, they are X and Y, that is two-dimensional. Three-dimensional means the column chromatography is a cylindrical. So that you can see that is, so that on the basis of physical state also. So by that also you can classify. So very simple, on the basis of interaction of the solute to the stationary phase. So the principle of different principle of chromatography, they can ask a question in a short form. The student should write, that is, what is the reaction between the solute, mean the sample, and the stationary phase. One is like adsorption chromatography, surface phenomena, adsorption, not absorption, adsorption. Partition chromatography between two immiscible liquids. Ion exchange, cation, anion exchange chromatography. Size exclusion, gel permeation chromatography. By means of molecular size, gel permeation chromatography. So, the principle of chromatography doesn't stop by means of adsorption and partition. It has ion exchange, size exclusion, that means molecular sieve, it will act as a molecular sieve, gel permeation chromatography, or size exclusion means gel permeation chromatography. By means of molecular size, it acts as a sieve to separate the compound according to the size of the molecular weight. So, on the basis of chromatography, bed shape, two-dimensional thin layer chromatography, TLC. Paper chromatography, a paper, X and Y, thin layer chromatography, also a plate with coated, that is thin layer chromatography. Three-dimensional chromatography, cylindrical, you can see the column chromatography. So X, Y, Z, it's a three-dimension chromatography. The movement of the molecules can be seen in X, Y and Z angle of, so that's why three-dimensional chromatography, column chromatography example. On the basis of physical state, liquid chromatography and gas chromatography. So, this gives an example for the student to write in the beginning stage a classification, a simple classification, different types of chromatography. We have not given any examples very detailed. Understand? And now adsorption chromatography, we want to explain about adsorption chromatography and partition chromatography, because whenever we talk about the principle behind chromatography, in all books they used to mention two major types like adsorption chromatography and partition chromatography. Very simple, student don't get confused. Adsorption, we know that what is adsorption, surface phenomena. It is not absorption, so it is reversible. A solid compound can adsorb, like charcoal, activated charcoal is an adsorbent. It adsorbs the compound and it can be released by means of using the other solvent, the same system, adsorbent. Adsorption is a surface phenomena, it needs a solid which can adsorb a liquid. So according to the affinity towards the adsorbent, what type of adsorbent you are using. So adsorption. Partition means, you know very well, partition means two immiscible liquids. Distribution of a solute between two immiscible liquids is called partition. Partition coefficient you might have studied in your smaller classes. The ratio between the amount of solute distribution between two immiscible liquid is partition coefficient. So that thing you should remember. Always. Adsorption means two things you have to remember. Adsorption will happen only the solid and a liquid, or a gas, it's also possible. Partition, it should be two immiscible liquids. Even a gas or a liquid, gas and liquid also is possible. But usually two immiscible liquids. Distribution of a solute between two immiscible liquids. The student should know very clearly. Don't do mistake. Understand? And even I will tell you the examples also very clearly. You can be very clear. Any book, anywhere you refer, you just stand in this point, you will not go in wrong. So this is an example just to explain about a chromatography. Basically, in an adsorption chromatography, how the compound is getting separated. This is a picture which I have taken, very interesting. To know about a mixture of protein as an example it is. And just an example with a diagrammatic example to make the student to understand. You see, there is a mixture of protein. It's an example. Even it can be a mixture of chemical compound, mixture of natural compound, your extract, which having mixture of compounds which you are interested to separate by chromatography. So this is a basic principle behind chromatography, how they get separated in adsorption chromatography. You see, this is a mixture of protein which is shown by an arrow I am showing, and this one we have one square I made. What is the interest of our experiment? You should know. What is the aim of our experiment? What is the main objective of our experiment in this chromatography is, you want to separate this moon-shaped red color protein which you are interested. You see that in the beaker which I have shown, the first arrow is, it contains the moon-shaped red color protein, a blue color protein, and a triangle of orange color protein also there. So there are three types of protein mixture is present in there, but you are interested to separate the moon-shaped red color protein, that is the interested protein which you want to separate. So how to protein? You can, so you need in this experiment a ligand. You can see a ligand which can only attract this red color protein, so that other protein will not be attracted by this ligand. So it will be eluted first. Eluted means it will be sent to the bottom of this tube faster. Other words, you can say it is not having affinity towards the stationary phase. Here the stationary phase which contain the ligand, which will hold this moon-shaped red color protein strongly. So the student should remember the words which I am using. Affinity, strongly, binding towards the ligand or the stationary phase. These all words you should remember. So see here, this is ligand and the protein which you are interested is moon-shaped red color, and you have shown the mixture of protein which contain all the three proteins. There is red color moon shape is also there, square shape blue is there, and orange shape triangle is also there. Can you see it? And now it is poured in the top of the column which contain a stationary phase. Now again remember, stationary phase means which is stationary in the glass tube, which is shown in the picture. If it is thin layer, a thin layer is stationary phase. If it is paper chromatography, paper is the stationary phase. Okay? So here the tube containing a stationary phase where the ligand is there. We are taking, it may be silica, it may be alumina, it may be, there are many stationary phase can be selected. But here, an example I want to say here, a ligand. Can you follow that? It's a polymer bed which is made there. So you see here, what happened? Next step, the same tube you are connecting with the reservoir in the top. Can you see the reservoir, the arrow which came just now? This is the reservoir which contain a solvent, common solvent. A common solvent or a common ligand which are used to make a flow through the column, through the stationary phase. So that one is called mobile phase. So now the student should know what is mobile phase, what is stationary phase, and what is elute. You see now, the mobile phase passes through the stationary phase and comes down. You can see there, the mobile phase or the solvent which you poured in the reservoir and coming down is taking only the triangle shape and the square shape protein. And the red color moon shape protein is not taken by the solvent which you poured in the reservoir which is made to pass through the stationary phase. Can you see that arrow? You can see the triangle shape and the square shape, blue color and orange shape proteins are eluted, removed by the solvent. by the mobile phase and it is collected. Can you see there? First one coming pure solvent. Second one pure solvent. Third one you can see the square shape blue color, orange shape, that is the orange shape triangle shaped, which is not having any affinity towards the ligand which is present in the stationary phase, which has been removed by the solvent which you have used. So it is collected in the third flask. Elute, it is called elute. Otherwise it is written in the picture, unwanted protein are washed through the column or removed through the column, or it is collected as an elute. Unwanted. Why it is called unwanted? Because our aim and objective of this experiment is we are interested in only one protein to be collected or separated from this mixture. That is that red color moon shaped protein which is shown in the picture, in the initial picture itself. Now you are changing the solvent. So you are changing the mobile phase. So this you will do in other system also. You will increase the polarity. You will change that is isocratic solvent system, or you can change, you can say that is many solvents, increasing polarity also can be used. So this solvent system, here the solvent we are using is ligand, the solution of the ligand which you are using in the next step. So what will happen? This ligand, the dotted, you can see just it is a picture only to show you the color, just to know, okay, ligand solution. Ligand solution, when you are adding, what it will do? It will bring all the interested protein, required protein, the red color moon shaped protein, which you need to make the complex with the ligand, which has the capacity, which is having the more affinity towards the mobile phase, which can be separated as shown in the picture. So this is how the separation takes place. Again, repeatedly I am telling, this you have to listen properly and you will know that many things you have come across the word stationary phase, mobile phase, elute, and the protein which you are interested is coming, and affinity I told, a compound which is having more affinity will stay for longer period, and the compound which is having less affinity will be eluted first. So the other proteins, uninterested proteins, are not having any affinity towards the stationary phase, towards the ligand which you kept in the column as a stationary phase. So they got excreted, or you can say they are eluted by the solvent faster. And finally, when you are using the ligand solution which is having the capacity to elute the protein which you are in interest, you can separate. So the 3 component mixture is an example how they separate by this adsorption chromatography. Adsorption takes place in the stationary phase by means of the affinity towards the stationary phase. One is having more affinity, they will stronger adsorb. Other is having lesser affinity, they will be loosely adsorbed, so that easily they can be eluted. So you see here, in its simplest form, this method of chromatography consists of passing a solution of mixture of compound needed to be separated through a hollow glass column which is as shown in the picture, packed with a finely divided adsorbent powder and collecting the solution that is called elute. This is just a column chromatography with colored one, just an example I want to show. Previously they used to even cut according to the color and they can find out, separate the compound also. The surface phenomenon is called adsorption, is utilized. The finely divided solids are capable of selective adsorption of other substances. So the adsorbent are selective. As you have seen in the previous picture, one protein is strongly adsorbed Other proteins are very fast, they are eluted. So selective adsorbent, which adsorbent you are selecting in a chromatography plays a major role for your separation. The component of a mixture introduced onto the column of adsorbent are more or less strong adsorbed. Those which are least strongly adsorbed, those which are less affinity, are carried down the column by passage of solvent and are fast to elute, who are not least, they are least strongly adsorbed, who are less affinity from the bottom of the column. First they will be eluted. The passage of more strongly adsorbed, suppose the compound is more strongly adsorbed, adsorption surface phenomena, this picture is just to show you the surface phenomenon, not it is absorbed inside. If it is more strongly adsorbed, the substance is slower. These are last eluted. Just now we have seen the picture. The moon-shaped protein was very strongly adsorbed because it has more affinity towards the ligand which you selected. So it slowly, finally it is separated. Last it is eluted. The fraction of eluent containing each component of the original mixture can then be separated and analyzed. This is about mobile phase. Solute means samples. A, B, that is red, green, just how they are adsorbed and distribution. Stationary phase is the ligand or the silica, gel or alumina. There are many components can be used as a stationary phase which will hold as an adsorbent. So alternatively, the entire adsorbent core may be extruded after bands containing the compound of the mixture have developed, and each band is removed by cutting the cylinder into sections. Individual bands can then be extracted with a suitable solvent to obtain each compound. This picture just to show you an example. We are going to study in detail about TLC, and now we are going to see about the column chromatography also. The initial chromatography technique is column chromatography. You see that how the color, because column we are running the solvent from the top to bottom towards the gravitational force, but in thin layer chromatography opposite, against the gravitational force. So you can see that is an example which they have shown. Orange, red, yellow. Here yellow, orange, yellow, red and orange. Then orange, red, yellow. Here you can see yellow, red and orange. Why? You can see that, as we have discussed, maybe the yellow is less affinity towards the stationary phase, so it is moving fast towards down. And the pink is moderate. Orange is more affinity, so slowly moving, so it is in the top. Same thing when you come to TLC, it is bottom to top actually, against the gravitational force. By capillary movement, the solvent, mobile solvent will move from the top to bottom to the top. So in that you can see the compound which is having more stronger affinity, that is orange product C, is staying in the down, and medium A, and which is less affinity moving faster in the stationary phase, which is taken by the mobile phase. So this is an interesting basic idea for the student to know about affinity towards the stationary phase plays the major role for the separation of mixture of compound in adsorption chromatography. Compound which is having more affinity towards the stationary phase of an adsorbent will stay longer time in the adsorbent. The compound is having less affinity will be easily detached from the adsorbent by the mobile solvent, and it will be eluted first. Very simple to Remember. Now we are coming to the partition chromatography. The basic principle. Partition means, I told, it needs two immiscible liquids or a gas and a liquid also possible. It is distribution of a solute between two immiscible liquids differs according to the solubility. Separation is based on the solute. Partition between two liquid phases or immiscible liquid phases. Relatively solubility by means of solubility basis. Partition chromatography. Partition chromatography is based on the difference partition coefficient. What is partition coefficient? Ratio of the solute. Distribution of the solute between two immiscible liquids is partition coefficient of the compound of a mixture which has to be separated between an aqueous or immiscible organic liquid. Simple. Water and a petroleum ether, or water and chloroform. They are immiscible. One is polar and one is less polar. So that distribution of the compound will be vary according to the solubility nature. The stationary phase is normally aqueous. Usually in a column, if you want to stay a liquid, you need some support to hold the liquid. Otherwise the liquid cannot stay as a stationary phase. Another liquid has to pass through the liquid. So we can use many means to make a liquid as a stationary phase. Another liquid has to pass through, and another solid substance which can hold the stationary liquid also needed for the technology wise you should think. The stationary phase is normally aqueous, which is mixed with an inert carrier powder to hold that liquid, pack in a glass column. Otherwise the liquid cannot stay. The mixture to be separated is dissolved in an organic solvent, is introduced into the column from the top, and the column developed with more solvent on different solvent of increasing eluting power. The column which is developed with a more solvent on different solvents of increasing eluting power. Example, if suppose you are having different mixture of compound, you have to use various solvent system which I have told in the classroom. In our previous class I was telling about the extraction procedures. To make your extraction itself, you should know what is the solubility of the component and what is the polarity of the component. According to the solubility, you should know the solvent system starting from hexane to water. How we know about the increasing polarity solvent system like hexane, petroleum ether, carbon tetrachloride, chloroform, acetone, ethyl acetate, ethanol, methanol, water, like that. You should know that what is the increasing polarity solvent system which can be used for eluting, increasing the eluting power of the mixture of compound can be separated. As shown in the picture, you can see a mixture of separated dissolved in the mobile phase is added in the column, and it will get separated as shown in the picture. Yellow, not yellow, blue, red, green, and how they are getting separated by the mobile phase through the process. And the component can be collected, that is called elute. Diffusion of the mobile phase through the stationary phase occurs. The different rate of travel of the constituents of the mixture are directly related to their partition coefficient, solubility between the mobile organic phase and the stationary aqueous phase. Water is the stationary phase, and if you are passing a non-aqueous solvent like petroleum ether, what all the components soluble in the petroleum ether will be eluted first. The compound which is insoluble in the low polar solvent will stay in the column with the aqueous phase. And the next, if you increase the polarity by means of changing it, the solvent system by benzene, slightly increasing the polarity. The compound which is soluble in the benzene can be separated. And suppose again if you are increasing the polarity by passing chloroform, the compound which is soluble in the chloroform will be eluted. Suppose again if you are increasing the polarity by methyl acetate, the compounds which are soluble in the methyl acetate will pass through the methyl acetate. When it is passing through the aqueous stationary phase, it can elute the compounds which is soluble. Even finally, if you want to do ethanol, and ratio can be also changed, polarity slightly can be varied by mixing the solvents, can also be done for separation of the compound. The finished chromatogram exhibit separate zones similar to those seen as adsorbent column. When you come to the column chromatography, in a column chromatography we used to pack column as a stationary phase. The column may be packed with solid or liquid. If the packing occur with the solid, the principle of separation is adsorption. While if the packing will be done by liquid, then the principle of separation is partition. Generally, in column chromatography, the most separation is based on adsorption principle. In adsorption column chromatography, the extract which is to be analyzed should be dissolved in the mobile phase and added into the column. The individual constituent in the extract will move with different rates according to the affinity, which depend upon their affinities towards the stationary phase or the adsorbents which you selected. The compounds which have more affinity towards the stationary phase will be separated in the last. It will be stronger binded and it will come separated slowly and it will be last. And while those with less affinity towards stationary phase will move faster and separated fast or first. The stationary phase which is used to fill the column should have uniform spherical shape or spherical size and high mechanical stability. Inert with solute or other components and mobile phase have free flow, freely available, inexpensive and able to separate the wide variety of compounds. Example, weak adsorbent is sucrose, starch, inulin, talc, while medium adsorbents is calcium carbonate, magnesium oxide, calcium hydroxide and magnesium carbonate. An example for strong adsorbent normally used for research is activated alumina, activated charcoal, Fuller's earth, silica gel 120, I have not mentioned, they all can be used as a column. The mobile phase may be used in individual or can be used in a mixture depending upon their polarity and the type of constituent to be separated. The column should be made of neutral glass or not affected by acid, alkali, mobile phase or stationary phase. The length of the column, usually the diameter ratio may be 10:1, 30:1, sometimes it may be 100:1 in ratio also available. So packing the column, you should know that very simple. A funnel or a burette burette is very small, little wider tube, column chromatography, which is available in our lab also. You need a funnel, glass column, eluent as we shown collecting sand, adsorbent, glass wool, collecting flask, which is marked. The lower portion of the column should be packed with glass wool, first initially, or cotton wool, to above which adsorbent should be placed. Selected adsorbent, silica gel, alumina, or whatever you are selecting. After packing the adsorbent, a paper disc is kept over it, can be placed over the adsorbent layer, so that on introducing the sample or the mobile phase will not disturb the adsorbent layer which is packed. There are generally two types of column packing techniques: dry packing technique, wet packing technique. When you come to dry packing technique, the column is packed with dry adsorbent and then flow the solvent or pour the solvent through the column till equilibrium is obtained. But the major drawback is that air bubbles are trapped between the solvent and the adsorbent, which produce trouble in separation of compounds when it is a solid adsorbent. The wet packing technique, the mobile phase is mixed with the adsorbent and poured into the column so that the air bubbles, air trapping can be avoided. So the air entrapment is comparatively less and uniform packing occurs in the column when you are going for wet packing technique. If the flow of mobile phase will be uniform, there will be no development of cracking of the column, and a good and a uniform separation of the extraction will occur. This is just a picture which we shown before, how the sample is loaded, stationary phase, and the sample is getting separated and the fractions are collected or eluting. Sample introduction, you can see usually the sample should be dissolved in at least a little quantity of the mobile phase and it can be added. The complete sample should be placed in the top of the portion of the column and get them adsorbed. And then the sample is isolated as an elution in the bottom as a small conical flask or test tube. Elution may be two types. Solvents, how you are using, one is isocratic, means same solvent for complete experiment. Composition of the mobile phase is same. Entire separation, even HPLC also, when you come to that, they are also having column, but it is very sophisticated, high performance column chromatography in minute micro molecules are separated. So entire system, if you are using a single solvent, it is called isocratic elution. Gradient elution, which we talked about, increasing the polarity by composition of the mobile phase, can be changed slowly to improve the separation. The polarity of the mobile phase can be changed by changing the mobile phase combination, as I told. Detection of components, previously by means of color, but detection, there are visual detection, can be done by various chemicals, can be sprayed, but in column chromatography we cannot spray. So the colored constituents can be directly visually colored bands moving. A column can be collected separately and it can be eluted. The colorless compound can be detected by UV techniques, refractive index detectors, monitoring by means of TLC, flame ionizing detectors can be also be used. In such techniques can also be used. Detectors can be used for detecting the components. The components can be recovered by different techniques. Sometimes even cutting the column into pieces, if it is a plastic column, it is easy for them to cut it and solubility can be found out, and by means of solvent several distinct zones can be cut, and the best method is elution, in which the component elute by mobile phase and the constituent is separated from the mobile phase. That is the usual thing which they are doing as a procedure. So this is simply a picture to show you the column. It's a glass tube. Actually, this can be even prepared by means of plastic tubes and cut into the shape, and they can do. Anyhow, this is a small class for a column chromatography and a basic thing which I want to make you to understand what is a chromatography. Chromatography is a technique which is used to separate mixture of compounds. And now I hope you should have at least an idea about adsorption and also partition chromatography and a basic example of a column chromatography. Thank you. We will continue in the next important classes like TLC, HPLC, paper chromatography. These all have to be cleared. Very interesting classes. Don't miss any class. Try to listen, write your notes, write yourself and submit, and also you refer yourself many books. Doubts should be cleared. If there is any doubt, then only you are in the study. Even I may also make some mistakes, correct in my mistakes, find out and just question is very important in any lecture. Thank you.